Hi everyone, my name is Elena and I've partnered up with Celine Kim for our final project, which was to observe the effects of UV exposure on yeast viability and fermentation. So for a little bit of background and introduction, S. cerevisiae, which is a strain that you all might be familiar with as we used it in class previously, often called baker's yeast, is a single-celled fungus used for thousands of years in bread making, beer brewing, and fermentation. So it converts sugar into ethanol and carbon dioxide through cellular respiration and fermentation. Because it grows quickly and is easy to culture and has well understood genetics, it is one of the most widely used model organisms in biology and genetics research. UV light is a form of ionizing radiation that carries enough energy to damage DNA molecules. When DNA absorbs UV, the energy alters nucleotide bases, especially thymine, and causes abnormal covalent bonds between adjacent bases. These are called pyrimidine dimers. These dimers disrupt DNA replication because the polymerase can't read the damaged section correctly. Cells try to repair the damage, but the repair process itself can introduce errors, aka mutations, or fail entirely, which can lead to slower growth, cell death, or loss of metabolic capabilities. Yeast needs healthy DNA to divide and metabolize sugar. When UV damage accumulates, fewer cells survive, leading to smaller colony counts. Survivors may grow more slowly, or fermentation output decreases because there are fewer active cells making carbon dioxide and ethanol. So, in bread dough or fermentation setups, this becomes more visible. For example, balloons inflate more slowly or less. I'll explain this more later on. And dough rises more slowly or fails to reach normal height. So our general hypothesis was that increasing UV exposure will decrease yeast viability and reduce fermentation output. How we were going to do this was in three steps. Step 1, UV mutagenesis and plating. Step 2, carbon dioxide fermentation measurement via balloons. And then step 3, visualization of dough rise. We used this specific brand of yeast. It was still S. cerevisiae. It was the rapid rise version. We used 6 different UV exposure times: 0, 15, 30, 60, 120, and 300 seconds. We used the serial dilution procedures that we used previously in class that you all are probably still familiar with. And then fermentation, we measured by balloon circumference every 15 minutes. Again, we'll explain more later. And we also measured dough height for visualization, obviously not consumed. Prior to starting all those experiments, we wanted to do a little bit like a little test for growth or some sort of reaction to confirm that this yeast is working in the way that we want it to. So on the left image, we see an example of the yeast hydration in YPD. And then 24 hours later, on the right, we see visible growth, some sort of reaction later on the right, which really gave us the green light for us to proceed. So part 1, UV exposure and plating. So we started with the yeast hydration in YPD. 0.5 grams of yeast into 12 milliliters of YPD, split it into 6 tubes, exposed it at different times to UV light, and then we plated them, incubated them 24 to 48 hours, counted colonies. And this is a really nice little sort of procedure schematic that Dr. Hartman drew out for us that really simplifies everything that I'm saying. And then this is a cute little time lapse that Celine and I filmed. This is us plating. I plated the 10 to the negative 5. She plated the 10 to the negative 4 dilutions. And
Plates.
this is the next image. And this is how we expose the plates to UV light. The top left shows a picture of all the plates. We put it into the incubator for, I believe it was 48 hours approximately. And then the bottom right shows the only 3 plates that really showed any colony growth, which expectedly were the 0-second plates, all 3 of them, which really is consistent with our previous knowledge and hypothesis. Part 2 was balloon fermentation. So we inoculated 100 milliliters sucrose medium with 1 milliliter yeast. or 2 milliliters, I'm sorry, it's a typo. And then balloons, that we sealed balloons for carbon dioxide output and the recorded circumference every 15 to 30 minutes for 2 to 3 hours. So the top left shows, again, sort of how we, like our little setup on how we expose the yeast suspensions to the UV light. And then this little time lapse shows me inserting the suspensions into the previously prepared flasks, and then Celine doing the more physically strenuous work of stretching the balloon openings over the flasks. So we took pictures in 15 to 30 minute to 1 hour intervals. We started at 11:45 a.m. when we saw absolutely no growth, and then 12 p.m., 12:30 p.m., we slowly see these balloons inflating, rising. By 2:30 p.m., or at even 1:30 p.m., they were all able to stand on their own. This is 4:40 p.m., and then at 5 p.m., you can see that there was a huge amount of inflation at the bottom of some of these. So we resorted to having to tape the bottom down so that all the carbon dioxide would only inflate the top of the balloon. And then we reinserted it into the incubator just in case we would observe even further growth in the next 24 hours. This was our data table. The reason I did not make a graph for this data set is because there was no specific correlation between the time in seconds the yeast was exposed to UV versus the circumference of the balloon, how fast it rose, etc. However, part 3, we did notice some sort of trend, and I'll get to that in a second. But part 3 was our dough rise visualization. I could not come in this day, so Celine did all the hard work. She made the dough composition and then added the mutagenized yeast, measured height over time. So this is another time lapse of her making, I assume, the suspension. Yep, there goes the YPD. And then making the mixture, subsequently making the dough, inserting the mutagenized yeast suspensions as well. Yes, lovely. And then with these 6 different flasks, beakers, sorry, she started out at 11:38 a.m. and then rechecked every 20 minutes, it looks like, around. And as you can see already, qualitatively, you can see some sort of general increase in the amount of dough that has risen. And then this on the top left, again, is our data table. I was able to format this data into a nice looking graph this time. However, the trends are really not what we totally expected, because it shows that for 0 seconds, for example, the navy blue, it shows less dough rise than the 300 seconds. In fact, it seems like dough height seems to be positively correlated, at least in our experiment, with increasing UV exposure time. So our original hypothesis was that increasing UV exposure will decrease yeast viability and reduce fermentation output. We did see partial support for this in the viability data. Higher UV time showed fewer surviving colonies, which means the radiation was damaging the yeast as expected. However, when we looked at fermentation performance, the results do not follow the same trend. In the dough rise experiment, even the most UV exposed yeast groups rose just as much, and sometimes even more than the control. That suggests that although fewer cells survived UV, the cells that did survive were still metabolically active and capable of producing carbon dioxide. The balloon fermentation data also does not show a consistent correlation, as mentioned before. Different UV groups inflated at different times, and the differences weren't systematic. That means we couldn't confidently say that increased UV exposure led to lower carbon dioxide production across the board. Some errors and limitations could have included uneven UV exposure distance, plate pipetting inconsistencies, balloon elasticity differences, dough mixing variations. There are so many variables that could have been unintentionally different across these 6 different times, which led to the unexpected results. So overall, our hypothesis was only partially supported. UV exposure did decrease viability, but it did not reliably decrease fermentation output in our experimental assays. And thank you so much for watching, and now I will open up the floor to any questions.